Hello dear students. In this video, we're going to speak about the photosynthesis in different climates. Let's start. Green plants thrive in environments ranging from hot and dry equatorial regions to freezing cold polar regions. Their success depends on their adaptability. To survive and breed, each plant has had to evolve specific adaptations to cope with the demands of its particular environment. These adaptations include ways of fixing carbon dioxide. C3 plants, fixing directly into the Kelvin cycle. C3 plants fix carbon dioxide directly into the Kelvin cycle as the 3-carbon compound glycerate 3-phosphate. Common and widely distributed, they include some of our most important crop plants, such as wheat, soybeans, and rice. C3 plants function efficiently in temperature conditions. However, they suffer two major disadvantages in hot, dry environments. First, to obtain sufficient carbon dioxide, C3 plants must open their stomata. Unfortunately, when stomata are open, they not only allow carbon dioxide to enter the plant, but also allow water to escape. So in hot, dry conditions, C3 plants have to either cease photosynthesizing or run the risk of wilting and dying. The second disadvantage relates to the ability of ribulose biophosphate carboxylase to combine with oxygen. Ribosco is the enzyme that catalyzes carbon dioxide fixation. On a hot, sunny day, carbon dioxide concentrations around photosynthetic cells decrease because a large proportion of the carbon dioxide is being used up on photosynthesis. In these conditions, ribosco combines with oxygen rather than carbon dioxide in a process called photorespiration. The process results in the loss of fixed carbon dioxide from the plant, reducing photosynthetic efficiently and plant growth. Unlike photosynthesis, photorespiration does not produce sugar molecules, and unlike respiration, it yields no ATP. As much as half of the carbon dioxide fixed in the Kelvin cycle may be released by photorespiration. Therefore, in hot, arid conditions or in conditions where carbon dioxide levels are low, C3 plants do not grow well. C4 plants, the Hatch-Slack pathway. C4 plants have evolved a special metabolic adaptation which reduces photorespiration. They do not use ribulose biophosphate to fix carbon dioxide directly into the Kelvin cycle. Instead, they use phosphoenol pyruvate to fix carbon dioxide as a 4-carbon compound, oxaloacetate. The reaction is catalyzed by phosphoenol pyruvate carboxylase. This enzyme cannot combine with oxygen. Consequently, C4 plants can continue to fix carbon dioxide even when its concentration is very low. The leaves of C4 plants are especially adapted to carry out this initial fixation. A ring of large closely packed cells called the bundle sheath surrounds the leaf veins. Surrounding the bundle sheath is a smaller ring of mesophyll cells. The distinctive arrangement is called Cron's anatomy and can be used to identify C4 plants. The initial fixation of carbon dioxide into oxaloacetate takes place in the small ring of mesophyll cells. Then the oxaloacetate is converted to malate, another 4-carbon compound. Malate is transported into the bundle sheath cells where it releases carbon dioxide. Once released, the carbon dioxide is reassimilated by RUBP and enters the Kelvin cycle in the same way as described for C3 plants. The metabolic pathway that transports carbon dioxide into the bundle sheath cells is called the Hatch-Slack pathway. As a result of this pathway, the concentration of carbon dioxide in the bundle sheath cells is 20 to 120 times higher than normal. C4 plants have two main advantages in hot, dry environments. First, because PEP carboxylase has a high affinity for carbon dioxide and does not combine with oxygen, C4 plants can continue to photosynthesize even when their stomata are closed for long periods. This reduces water loss and photorespiration. C4 plants need only about half as much water as C3 plants for photosynthesis. Secondly, because high carbon dioxide concentrations can be maintained in the bundle sheath cells, C4 plants can increase their photosynthetic efficiency. These adaptations enable C4 plants to outcome C3 plants in hot and very sunny conditions, but not in temperate conditions. Fewer than 0.5% of plant species are C4 plants, yet they include economically important crops such as maize, sugarcane, and millet. Cam plants photosynthesizing in the desert. Although C4 plants are well adapted to occasional periods of drought, they cannot cope well with desert conditions. A group of plants, including cacti and pineapples, have evolved a third type of carbon dioxide fixation which enables them to survive in very dry climates. These plants are called cam plants. CAM is an abbreviation for crassulacean acid metabolism, a type of metabolism first observed in the family of plants called Crassulaceae. Cam plants conserve water by only opening their stomata at night. During the night, they fix carbon dioxide into oxaloacetate, which is converted into malate. This acts as a carbon dioxide storage compound. During the day, malate releases carbon dioxide into the Kelvin cycle. This allows photosynthesis to take place on hot, dry, sunny days, even though the stomata are closed. Cam plants conserve water very well and are able to survive in extremely dry conditions, but cam plants do not photosynthesize very efficiently. Most are very slow growing. Where there is plenty of water, cam plants cannot compete well with C3 and C4 plants. Cam plants and C4 plants have a similar metabolism. Carbon dioxide is first fixed into a 4-carbon intermediate before it enters the Kelvin cycle. However, in cam plants, the initial fixation and the Kelvin cycle occur at separate times, whereas in C4 plants, the initial fixation and the Kelvin cycle are separated structurally, but both occur during the day. C4 plants live in hot, very sunny, and periodically dry environments, but where lack of water is rarely a limiting factor and annual rainfall is high. Cam plants are desert plants that live in areas of very low annual rainfall. Note that C3, C4, and cam plants all eventually use a Kelvin cycle to make glucose from carbon dioxide. That is the end of our video session. Next time we're going to speak about the leaf structure. Thank you for watching.